Alright, so in this video we're going to be talking about proteins, mostly about protein structure. In previous videos we had talked about amino acids and how amino acids can be combined into peptides, but as those peptides become larger and larger and larger, then they become, they're considered to be proteins, and proteins will fold up into specific shapes and perform specific functions. And so protein shape is usually defined as either fibrous or globular. And a fibrous protein tends to form long fibers, and they include proteins called such as keratin and collagen that make up connective tissues in the body, or make up keratin specifically is found in like skin and hair, fingernails. So the fibrous proteins tend to be structural proteins, the very tough type of protein that holds tissues together. Whereas globular proteins tend to be roughly spherical in shape. They include proteins such as hemoglobin and most enzymes. And they tend to be water soluble, whereas the fibrous proteins tend to be insoluble. But we further classify protein structure into 4 different levels. The primary, secondary, tertiary, and quaternary. And we'll discuss what each of these levels of protein structure mean. And we'll begin with the primary structure first. So primary structure is simply the sequence of amino acids in the protein. That we go from the N-terminus to C-terminus. And just like letters in a word, amino acids in a protein, that we can rearrange the amino acids in the protein to come up with different proteins. So the order of amino acids matters, just like the order of letters in a word matters. And again, just like letters in a word, we only have 26 letters in the alphabet, but we use those 26 letters to make millions of different words. And we only have 20 amino acids, but we can use those 20 amino acids in different orders, different combinations, to make millions upon millions of different possible proteins, depending on how we arrange the different amino acids. And so the primary structure of a peptide is simply the sequence of amino acids in that protein. And we can think of it as like how we spell a word using letters. We can build the peptide out of amino acids in the same way. So the secondary structure is caused by how the peptide backbone folds on itself. And how it's held together through hydrogen bonding. And so as we've drawn all of our peptides essentially as straight lines, going from one amino acid to the next. But as those peptides become longer and longer and longer and longer, then they tend to fold in on themselves, and they can adopt some pretty complicated shapes. And the shape is important. In fact, it's necessary for protein to function. And we essentially the all of the information required to build the protein, to fold it into the right shape, is found in the primary structure. That the different identities of the amino acids as we go from one end of the say the N terminus to the C terminus of the peptide cause it to fold up in a specific way to make a specific shape so that it can perform a specific function. And the two common secondary structures that we'll look at that have to do with this peptide backbone, and I should specify what. Let's draw another peptide so that we can take a look at what is the backbone and what isn't the backbone. So I'm drawing the peptide backbone in blue. So I've got 1, 2, 3, 4, 5 amino acids here. If I left it as is, we could say that it's glycine, 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 glycine. But we'll draw the side chains on these amino acids in red. So here's a serine, and I'll divide the. I'll show where the peptide bonds are. Okay, so the first one is serine. Then the next side chain would make this in alanine. The next side chain this would be lysine. Then cysteine. And then aspartic acid. And so again, this backbone is drawn in blue, and the side chains are all drawn in red. And secondary structure is caused by this backbone folding up into a certain shape, and it's held together through hydrogen bonding because this hydrogen atom on the amide can act as a hydrogen bond donor, and the carbonyl oxygen can act as a hydrogen bond acceptor. And so we can get a hydrogen bond from this hydrogen atom to one of these carbonyl oxygens, and it won't happen immediately adjacent like I've shown here, but if we have a really long backbone, then the carbonyl from one and the hydrogen, the carbonyl from one amino acid and the hydrogen from a different amino acid far away can form a hydrogen bond and hold that structure together. And we'll take a look at a couple of examples on how a couple common examples of secondary structure caused by folding of the protein backbone. So the first one that we'll look at is called an alpha helix. And it's caused by when the peptide backbone twists into a spiral shape. And I will attempt to draw it. That doesn't look very good at all. All right, so that's a little better. We'll shade in the back part of the yeah. So now we have this spiral shape, this alpha helix, and it's held together by hydrogen bonds. And so again, we have the carbonyls from the carbonyls from one part of the peptide backbone, and the hydrogens from the other part of the peptide backbone that would form the hydrogen bonds that hold all of this structure together. They tend to be very flexible and act like springs. And so you can see how it's spring-shaped, and it can be compressed and stretched and make for a more flexible part of the protein. So the other secondary structure that we'll look at is called a beta sheet. And it's caused by, if we take that peptide backbone and we stretch it out and we place it next to another peptide backbone, then those two peptide backbones can hydrogen bond in between, and it'll make a sheet-like formation. So to help illustrate that, we'll draw out a peptide backbone. We'll ignore the side chains. And it would go on much further than what I've drawn. And then we'll draw a second peptide backbone see if I can't arrange this a little better. Okay, so we have two peptide backbones, and we can get hydrogen bonding from these hydrogen atoms to these carbonyls, and vice versa. And so when we get lots of peptide backbones held together by hydrogen bonds, it can form a sheet-like structure that it's not so flat like a sheet of paper. It's kind of a zigzag shape, like a folded piece of paper. And these folds then, or these sheets can be arranged in two different ways. If the peptide backbones all face the same direction, and by direction I mean as we go from the N-terminus end to the C-terminus end, then we call that a parallel sheet, the parallel beta sheet, because everything goes in the same direction. And if these are all part of the same protein, which they usually are, then they could be connected, that the backbone of the, we could say that the N-terminus of the protein is up here, and the backbone comes along, and then kind of loops around behind, and then starts the next part of the beta sheet, and again loops around behind, starts the next part of the beta sheet, and then we get to the C-terminal end of our protein. Or the other way they can be arranged is they can be anti-parallel. And that at least tends to be a little easier to draw. Because again, the direction as we go from the N-terminal end to the C-terminal end, the sheets are now pointing in opposite directions. And so we call this anti-parallel because as we go from the N-terminal end to the C-terminal end, the direction of the peptide backbone changes. So anyway, the alpha helix and the beta sheet tend to be the major examples of secondary structure, and most proteins are built from combinations of alpha helix and beta sheet structures, that the peptide backbone is folded up into these shapes, and then these shapes will combine into the larger structure of the protein, which we'll get into next as we discuss the tertiary structure. And that is the overall three-dimensional shape of the protein. and that it tends to be held together by interactions of the side chains. So the secondary structure is held together by interactions with the peptide backbone. Tertiary structure tends to be held together by interactions of the side chains, not the backbone. So when I drew, I won't draw the whole thing again. When I drew the backbone in blue and the side chains in red, tertiary structure is held together by interactions between the side chains of different parts of the protein as it folds up. And we'll discuss that in more detail. If you remember that I had previously said that proteins could be either fibrous or globular, well, globular proteins tend to have a tertiary structure that puts their hydrophobic amino acids, so your nonpolar side chains, into the middle of that glob, where the hydrophilic or your polar amino acids they face the outside where all the water is. So this is what makes a globular protein water soluble. Is we'll draw a spherical protein that that peptide backbone is going to like fold up into a specific shape, and we'll draw the hydrophilic side chains in blue. These like to be around water. They can form hydrogen bonds with the water, and so they'll be on the outer surface. And the core of the globular protein will be made up of hydrophobic side chains that I'll draw in red. And they don't like water, so it folds up to hide these hydrophobic side chains away from the water. And then I'll just draw some water around the outside. So the shape of the globular proteins tends to be dominated by this rule that the hydrophobic nonpolar amino acids get packed into the inside of the protein, and your hydrophilic polar amino acids will be on the outer surface where they can interact with the water. And so non-polar in, polar equals out. And these globular proteins also tend to have pockets on the outer surface. So we'll draw in a pocket, and small molecules tend to bind into those pockets. So things that what we might consider normal organic molecules, rather than large protein molecules, the small molecules can bind into these pockets, and we call those pockets binding sites. And these will become important when we start discussing enzymes. And so the tertiary structure, now we'll get into a discussion of what exactly holds this tertiary structure together. I had mentioned that the tertiary structure is held together by interactions of the side chains. So we'll list out what those interactions are. We have non-covalent interactions, such as hydrogen bonding, ionic interactions, where a positive charge is attracted to a negative charge, and we oftentimes call that a salt bridge. That's just a term used by biochemists to describe the attraction between a positive part of a protein and a negative part of a protein being held together through electrostatic interaction. And we have the hydrophobic effect, where two hydrophobic amino acid side chains are held together because they don't like to dissolve in water, and so they come together to help squeeze out the water in between, so there's less water around, and they stick together through London forces. And then we have covalent interactions, disulfide bonds. That in the amino acid video I mentioned that two cysteines, which have thiols, that if you take the two sulfur atoms of the cysteines, you can stick them together and form a disulfide bond that holds the two cysteine amino acids together. And so, as an example, we'll draw a pretend peptide. So we have a couple of polar amino acids. We have serine and serine and tyrosine that they can form a hydrogen bond between the hydrogen and the oxygen. So that's one interaction. Oh yes, while I'm here, I'll say that these are all non-covalent, and the disulfide bonds are covalent. So anyway, moving back to our pretend protein, we can have the salt bridge. We'll draw an aspartate, which is negatively charged, and I'll draw a lysine, which will be positively charged, and that negative charge to positive charge can form an ionic bond that holds these two amino acids close to each other, and that will be a salt bridge. We'll draw a couple hydrophobic amino acids. We'll use phenylalanine and valine, and so these will stick together because they don't like to be around water, and when they stick to each other, they'll remove that water that was in between. So they'll squeeze that water out, and so that's an example of the hydrophobic interaction. And finally, we can have a pair of cysteines where their sulfur atoms are covalently bonded to each other, forming a disulfide bond. And so these disulfide bonds tend to be pretty tough, since it's a covalent bond and everything else is non-covalent. And so this covalent bond is much more stable, but it's not irreversible. That in the presence of reducing agents, this bond can be broken, and inside the cell we have a lot of reducing agents. And so disulfide bonds on proteins found inside cells tend to be very rare. But proteins that are excreted from the cell, outside of the cell, tend to have a lot more disulfide bonds. So that helps to hold the proteins together, helps to preserve their tertiary structure. So there's another aspect of tertiary structure that we should talk about, and that is the prosthetic groups, which are non-peptide molecules bound to proteins. I had mentioned the proteins can have binding sites, and sometimes small molecules will fit into those binding sites so tightly that they're almost considered part of the protein. That in fact they're necessary for protein function, but they're not made up of amino acids. And a common example of a prosthetic group is the heme group. And heme is found in hemoglobin and myoglobin and several different enzymes because it can bind to oxygen. And so, like hemoglobin just uses that to carry oxygen throughout the blood. Enzymes might use it so that they can add oxygen to other molecules. The heme group see if I can draw it okay. And then I have to add all the other bonds. And then it's got some atoms around the outside. So you don't need to know the exact structure of this heme, but what allows it to function by what allows it to bind to oxygen is the presence of this iron ion located in the middle of this ring. That it's held in place by electrons from all of these nitrogen atoms. So the nitrogen atoms all have these lone pair electrons, and so they are attracted to this positively charged iron ion, and that holds that iron ion in place, and then oxygen. So I'll draw an oxygen molecule will can also bind to the iron, and so we're drawing it from the top down. If I rotate it, then essentially it's a flat disc with the iron in the middle, and I'm not drawing this to proper scale, so the sizes aren't exactly accurate, but the oxygen can then bind to the top, and it's held to the protein by a histidine. And so the nitrogens in the histidine amino acid will also bind to the iron, and so we'll get a binding pocket around the heme, so that the protein holds this heme group into a specific site on the protein, and oxygen can come in and bind to that iron, and then the protein can either carry that oxygen to another part of the body where it's needed, or it can take a small molecule. I'll just try to draw a random small molecule here. That's totally fake, but you get the idea that the oxygen can be used to react with that small molecule, and in a lot of metabolic enzymes, especially what's called the cytochrome P450s, and there are hundreds. It seems like there are hundreds of these cytochrome P450 proteins that are involved in the metabolism of different molecules inside the liver. That they take the oxygen and they react it with the small molecule in order to help digest those molecules. So anyway, that's probably more than we need to know about prosthetic groups and heme right now. But that's the tertiary structure of the protein, and so we'll move on to quaternary structure. And quaternary structure is a combination of more than one peptide backbone into a single protein. And it can get a little confusing because sometimes we call these different peptide backbones to be proteins, so we have more than one protein coming together to form a single overall structure that is also a protein. But we call these individual peptides, the individual parts are called subunits. And the subunits are held together the same way the tertiary structure is held together, through the non-covalent interactions, which include hydrogen bonding and hydrophobic effects and salt bridges, but also through disulfide bonds. And a good example of quaternary structure is hemoglobin, because it's made up of 4 parts. It has 4 subunits, 2 alpha and 2 beta subunits. And so if I were to draw this out, alpha, alpha, beta, beta. And in the case of hemoglobin, each of these subunits also contains a heme, which I won't draw the entire heme structure again, but each heme each subunit contains a heme, and each heme can bind 1 molecule of oxygen. So that means that a single hemoglobin unit can carry 4 molecules of oxygen, because we have the 4 subunits, and each subunit carries 1 molecule of oxygen. So another important example of quaternary structure is collagen, which again is found in connective tissue. And collagen is made up of a triple helix. And I'll see if I can draw a reasonable triple helix. And so we have these three different collagen peptides that all wrap around each other into this helical shape, and when the collagen is newly formed, there's no connection between the different proteins, that they're held together through non-covalent interactions. But as the proteins age, then we start to get covalent bonds between the different strands, and so that makes the helix even tougher to pull apart, because now we'd have to break all these covalent bonds too. And so that helps to make collagen a very strong protein, and that's why it's found in connective tissues that need to be held together very tightly. Another good example of quaternary structure are antibodies. And antibodies, of course, are found in the immune system. That they have binding sites that allow them to recognize and bind to specific structures called antigens. And so if we draw the antibodies are kind of like a Y-shaped molecule made up of 4 different proteins. And those 4 proteins are held together through disulfide bonds. And so the black part is referred to as the heavy chain, and the blue is called the light chain. And so 2 heavy chains and 2 light chains come together to form the antibody. And this part, the ends of the Y are the binding domain. That this is where the antigen will bind to. And so if we have like let's say a bacteria, and that bacteria is going to be covered in proteins, proteins and other molecules. So I'll draw some of those. We've got our red proteins and our green proteins. And some blue proteins. But anyway, the antibodies all have different binding domains, and so some of these binding domains, because they're different, they bind to different antigens, and they bind with a very high affinity. So once they bind, they are highly unlikely to ever let go. And so an antibody can come in, and let's say that it binds, that it has a binding domain that recognizes part of the red protein. And so the antibodies would come along, and that they would bind to the red proteins. And so in this case, the red proteins are antigen. And then once the bacteria is covered in antibodies, then your immune system cells can recognize that this is a foreign substance, that it's been coated with these antibodies that recognize the specific bacterial antigens, and come in and attack that bacteria. And that's how your body recognizes different pathogens, viruses, and bacteria. So we've been talking a lot about protein structure, your primary, secondary, tertiary, and quaternary. Now we'll talk a little bit about what happens when that structure goes wrong. That we have what's called the native structure, and that is the biologically active, normal, like the normal structure of a protein. We call that the native structure. This is the shape that a protein should have. And denaturation is any change that disrupts normal shape and therefore function. And so the denaturation is usually causes a loss of biological activity. It's usually irreversible, and it can be caused by changes in temperature, or pH. There are other things that can cause denaturation as well, but these tend to be the most common. That a good example of denaturation that you've probably all seen is cooking eggs. When you take the eggs and you heat them up, the liquid proteins in solution, they denature and they aggregate together to form the solid cooked egg. You cannot really uncook an egg; that it's irreversible. But you could also sort of denature those proteins if you add like a strong acid to the proteins. That it would also cause them to denature. The addition of soap or detergents can also disrupt the hydrophobic interactions that hold proteins together, and so that would cause them to denature as well. Now, an interesting an interesting topic related to denaturation are the prions. And prions are an unusual type of denatured protein that causes normal proteins to also become prions. And so once it becomes a prion protein, these prion proteins tend to aggregate together, and it disrupts normal cellular function, and it causes disease. And so if we start with a healthy protein, a normal protein, and it becomes denatured somehow, well, then it might become a prion. So I'll label normal, and if it becomes denatured, and if it's denatured in just the right way, and this doesn't work with all proteins, only a very small number of proteins can be denatured like this. But if it goes from normal to prion, then that prion can take other healthy proteins, and it can convert those healthy proteins to other prion proteins. And each one of these prion proteins can go and convert healthy proteins to more prion proteins. And so it's a chain reaction that builds and begins destroying healthy tissue, oftentimes found in the brain. That diseases caused by prions include mad cow. Creutzfeldt-Jakob disease is essentially mad cow disease found in humans. I can't spell Creutzfeldt very well, so I'm not going to try to spell it. Another one found in humans is Kuru, found in cannibals who eat human brains. Oftentimes will get sick with Kuru. Scrapie, found in sheep, and chronic wasting disease, found in deer. They're all caused by prion proteins. That these proteins, if you were to eat them, then some of these proteins might make their way to your brain, begin converting healthy proteins to prion proteins, and essentially destroy your brain tissue. And so it's an unusual case of what can happen when protein structure goes wrong. So that's our discussion on the structure of proteins. We'll discuss enzymes in the next video.